In pharmaceutical analysis, assay concentration, also known as potency, measures the exact amount of an active ingredient in a drug product or sample. It's crucial because it ensures that the medication has the correct concentration of the active substance, which is essential for achieving the intended therapeutic effect. Here's an in-depth look at assay concentration, its calculation, and its role in the pharmaceutical industry. Definition of assay concentration, also known as potency. Assay concentration or potency represents the amount of active pharmaceutical ingredient or API in a given sample relative to what is stated on the label. This measurement ensures that each dose delivers the correct therapeutic effect to the patient. Potency can be expressed as a percentage of the labeled amount, or as milligrams, micrograms, or other units of measurement, depending on the drug. Formula: assay concentration equals measured active ingredient divided by labeled active ingredient. That shall be multiplied by 100. For example, if a tablet is labeled to contain 100 milligrams of an API, an assay determines how much of that 100 milligrams is truly present. If testing reveals 98 milligrams, the potency or assay concentration is 98%. Importance of assay concentration. Assay concentration is critical for several reasons. Therapeutic efficacy ensures the correct amount of the active ingredient is present, so the drug can deliver its intended effect. Safety prevents overdosing or underdosing, which could lead to adverse effects or ineffective treatment. Regulatory compliance. Regulatory agencies like the FDA set strict potency standards for drugs to ensure patient safety. Quality control. Regularly measuring assay concentration is part of maintaining batch-to-batch consistency and product quality. Calculating assay concentration with examples. Here's a basic example to understand the calculation. Example 1. Percentage assay calculation. Suppose a tablet is labeled to contain 500 milligrams of API, but upon analysis, it's found to have 490 milligrams of active ingredient. Assay concentration equals 490 milligrams divided by 500 milligrams. That shall be multiplied by 100, which equals 98%. This means the tablet has a potency of 98%, indicating it's slightly below the labeled amount, but may still fall within acceptable limits. Example 2. Potency by concentration in solution. In cases where the API is dissolved in a solution, assay concentration is expressed as a concentration per volume. For instance, if an injectable solution is labeled to contain 10 milligrams per milliliter, and the assay shows 9.8 milligrams per milliliter, assay concentration, in percentage, equals 9.8 milligrams per milliliter divided by 10 milligrams per milliliter, that shall be multiplied by 100, which equals 98%. This indicates the injectable solution has a potency of 98% of its labeled amount. There are several analytical techniques used to measure potency, each offering accuracy and specificity depending on the drug and its formulation. Some common methods include high-performance liquid chromatography, or HPLC, is a widely used technique that separates the active ingredient from impurities and quantifies it accurately. UV visible spectroscopy is often used for simpler formulations. It measures the absorbance of a sample at specific wavelengths to determine concentration. Mass spectrometry, known as MS, is used for complex mixtures. MS identifies and quantifies the active ingredient by analyzing its mass-to-charge ratio. And lastly, titration is a chemical method used in cases where the API reacts with a reagent, allowing quantification based on the amount of reagent consumed. Each method has its advantages and is chosen based on factors such as sensitivity, specificity, and the type of pharmaceutical product. Factors affecting assay concentration. Several factors can impact assay concentration. Stability of the active ingredient. Some APIs degrade over time or under certain conditions, which can reduce potency. Manufacturing variations. Differences in manufacturing processes can lead to slight deviations in API concentration across batches. Storage conditions. Temperature, humidity, and exposure to light can affect potency, especially for sensitive APIs. Formulation components. Certain excipients, which are inactive ingredients, may interact with the API, impacting assay concentration. Understanding these factors helps in designing more stable and effective pharmaceutical products. Acceptable assay range in pharmaceuticals. Regulatory agencies typically require drug products to meet specific assay concentration ranges to be considered acceptable. For example, oral tablets and capsules usually have a potency range allowed between 90% to 110% of the labeled amount. Injectable products often have stricter limits, usually between 95% to 105%, due to the direct delivery to the bloodstream. The acceptable range varies depending on the drug, formulation, and regulatory standards, but consistently achieving these standards is essential for product approval and patient safety. Assay concentration versus percentage purity. While assay concentration or potency and percentage purity are related, they measure different aspects. Assay concentration measures the amount of active ingredient compared to the labeled amount, ensuring the right dose. Percentage purity measures the amount of desired substance relative to impurities, ensuring the sample's overall quality. For example, a tablet might have a potency of 100%, meaning the correct dose, but a purity of 95%, indicating that 5% of the sample is made up of impurities. Both are crucial but serve different quality control purposes. In summary, assay concentration or potency is a vital measurement in pharmaceuticals that ensures each drug dose contains the intended amount of active ingredient. Accurate assay measurements are essential for therapeutic effectiveness, safety, and compliance with regulatory standards. By consistently monitoring potency, manufacturers can guarantee high-quality medicines for patient care.